Hello, everybody. It's Mr. Caudale. I hope you're doing well. I'm doing well. It's the first time I've recorded one of these lectures while wearing pants. That must suggest to you, like it's become clearly evident to me, that the seasons are changing. We are now in early October. You're probably watching this sometime in January or February. I hope everything's okay. I really do. Um, I like evolution, and we've been going through it, and I hope you like it too. Some would argue that this is one of the most important chapters in the textbook because the whole idea is that we've put together all these pieces of evolution, and now it's all about macroevolution. The idea is that if you take the pieces of evolution and you put them all together and you cumulatively stack them, that there is no other conclusion you can make than the inescapable one that all life originated from a common ancestor, and that was a single-celled organism. Now, that's what your book wants you to think. I'm not even telling you if I believe that, but you don't have to believe that. I don't care. But you do have to have an open mind, and you are responsible for this material. So, even if you don't believe it, I think it's sexy to know both sides of any argument. It makes you sound more intelligent, and then you'll know what your opposition is thinking. I'm just saying generally, but keep an open mind. Look at kind of the big picture here. We're just going to do 25.1. It's just a couple of pages in our textbook in the 10th edition, and then we'll go from there. But first, like usual, a movie. How could you not be excited to dive into chapter 25 after that? I spent so long making, just kidding. Look, I'm going to be honest. The story of life is tremendous. And I do believe that life has originated on this planet over the last 3.5 billion years. There's a little magic sprinkled in there, probably. That's what I believe. But the point is that the whole process of evolution and now this biological superstructure that has been created it's just remarkable when you sit back and you wrap your head around it. I'm going to step over here because I have to mention this. Being a biologist, the reason why I got into this field and was so excited to be a biology major whenever I was there in the 90s was because of life and how cool it was and how everything was interconnected and how elegant all the processes were. And it just blew my mind at night studying in the library. And I just couldn't even fathom it. I wanted to learn more. And then I eventually wanted to share what I knew with others and try to spark their imagination too. But I have to say, we suck right now as a species. And I hope when you're watching this that things are getting better. At this moment, I'm not going to tell you what year it is or what month it is. I'm disappointed with humanity, and I am as guilty as anyone else. But things have to change, and we have to preserve life on this planet. There was just a report that came out the other day from the World Wildlife Fund that 70% of all populations of animals have gone away over the last 50 years on this planet. The species still live, but their populations are much smaller of birds and mammals and insects than they used to be. It's really sad. We have got to protect this planet. That's my 2 cents. Please be an advocate for life as you go forward in yours. This is all we're going to do. 25.1. So you have to make 2 major assumptions here. Unbelievable amounts of geologic time that you can't even wrap your head around. If I was to say, oh, that happened 171 million years ago, or I said, that happened 173 million years ago, would it make any difference to you? But that's 2 million years difference. My point is these are spans of time that you can't even fathom. And number 2 is chance. The whole evolutionary philosophy is based on chance, that if you have enough chance interactions of molecules or organisms together in an environment or something, or DNA mutations, that you're going to eventually get something that might be advantageous in that environment. So open your mind to these concepts. You don't have to believe them or not, but open your mind over the next few units of this chapter, please. Well, we can look at fossils. That's really basically what we have when we try to piece together what has happened on this planet. We can see things such as cell formation that they go over pretty well. The most old fossilized cell beginnings that we have, and you'll see that in the next portion of this chapter. We then can identify when photosynthesis took place, and then the atmosphere on our planet began to change to an oxygen-based atmosphere. A vertebrate is an animal that has a vertebral column, and the only reason why you'd have a bunch of bones in your back with a hole down the middle is to protect a spinal cord, and that suggests higher beings where they have some sort of like massive tissue. This is neuronal tissue in the nervous system, and then that has kind of fanned out in the rest of the central nervous system, which in our case would be our spinal cord. And it's going to be able to deliver messages and then receive inputs from distant parts of the organism, like our spinal cord going out to our limbs. So we can see the emergence of these guys on land and in the ocean, of course, through fossils. And then there's been many mass extinctions, and we're going to talk about those later in this chapter. And they're really important. Well, this was about 4.5 billion years ago that this went down. And what I mean by that is the formation of our planet. And so where did we get all of the carbon, hydrogen, oxygen, and nitrogen, phosphorus, and sulfur that most of life is made up of? Well, it basically is stardust, and it congealed into this big ball that we now call Earth. It began to be bombarded by meteors and asteroids for sometimes hundreds of millions of years, and scientists believe that those brought a lot of the water that we have on our planet, as well as a lot of the more obscure elements that we have on our planet, and more carbon and hydrogen. And so it just fed this ball of rock, essentially, which was cooled to become hard with a bunch of atoms. And so the idea is that those are going to get together. In the atmosphere of our planet, we had water vapor. And I want you to press pause and think about what you think was in our planet's atmosphere. Way back when. We're talking about, this is about 3.5 billion years ago, right before life began. Okay, I'm back. I'm going to tell you that there was oh, that's not how you spell water. This is how you spell water. There was certainly water vapor. There were gases such as nitrogen gas, N2. There was a derivative of that we call the ammonium ion. There was carbon dioxide gas. There were hydrogen sulfide gases, and there was hydrogen gas. Well, what do you not know? What do you notice is missing here? What do we like today? What do we like in our atmosphere? We like oxygen. And I'm going to write down here, no oxygen. That's the key point here, is that there was no oxygen. And in order to burn glucose or these other carbs or other foods that we like to burn in the mitochondria of our cells, we need oxygen in order to do so. You have to remember, this was a very toxic place, a toxic environment. There was no oxygen present for a long period of time. My memory serves me correctly. For like 1 billion years, I think there was no oxygen in the environment. As a result, I think things had to stay small. They weren't probably very efficient at once life had begun. They weren't efficient at breaking down food and making energy from that food. Oxygen was the key player here, and so we probably just had single-celled organisms that were on our planet. Well, one of the individuals that was really interested in studying this was Stanley Miller, and this was in 1951. He was a student at UC San Diego, and he was also at the University of Chicago, and he said, well, I'm going to mimic the Earth's early atmosphere, and I know that you were introduced to this in chapter 5, but it's so important they revisit it in chapter 25. Actually, that was chapter 6. Chapter 25, they come back to it. So we've got this apparatus here, this piece of glassware, and he has some water, and he's boiling the water, mimicking the early Earth's oceans. He's got sparks going into the apparatus, which is mimicking lightning strikes. He's got all these gases that may have been present. And his whole idea was he was just going to let this thing run for a while, weeks and weeks. And then he was going to look at the bottom of the sample for chemical analysis that you see there on the screen or on your screen. And then he was just going to look and see, hey, could the earliest macromolecule precursors, the beginnings of carbs and lipids and proteins and nucleic acids, could they form in this environment? So remind your neighbor about what he found, please. Here's a 30-second movie on some of the stuff that he found.
I was in Texas with my colleague Antonio Lascano, who's also a good friend of Stanley's, and we were preparing to give a couple back-to-back lectures, and we were looking at each other's slides. Antonio just sort of mentioned to me, he says, well, you know, Stanley showed me a vial of stuff that he said he'd saved from his original experiment. And I said, you're kidding me. I said, he never mentioned this to me. And he goes, well, he just sort of showed it casually and put it back in the box. I said, where is that box? He says, well, I don't know. It was in his office. Well, all of a sudden I realized I'd inherited everything that was in his lab and office. And so when I got back to my lab, we started looking around, and here we found these old cardboard boxes marked 1953, 1954 experiment. We started opening them, and in that we found extracts from each of the 3 experiments, including this one that we call the volcanic sudden I realized here we had the original samples in our hands that we could now reinvestigate that experiment using modern analytical methods. And Stanley did not have it at his disposal at the time. And what we found was indeed that the volcanic apparatus not only produced a richer mixture of amino acids, but many of the amino acids are ones that have never been detected previously in a simulated earlier synthesis experiment. So it was really revolutionary.
It's pretty exciting that Dr. Banda could go back and see all the stuff that had been created now with new modern analytical techniques. And I think it's also exciting that you can put all these gases and some water and some sparks into this glassware, and with enough time and enough chance, you can get amino acids. You could get the beginnings of lipids. And so this is the argument that this is what happened in this bubbling cauldron of the primordial ocean on our planet. I will tell you before we get to this question that's on the screen, I read an article this morning about Dr. Banda and Dr. Miller. They did another experiment where they put cyanide and ammonia together, and then they put it into a deep freezer. This was at a negative 108 degrees Fahrenheit, supposed to mimic the temperature on Jupiter's icy moon Europa. And so the idea was, hey, maybe we can mimic the conditions on Europa, although they don't really know what the conditions are. They left it in this freezer for 25 years. So they yanked it out. They then analyzed what was in there, and sure enough, those things had come together to make amino acids. All those atoms come together if you give them enough time and enough chance, and eventually you're going to find what you're looking for. And the idea then in all these instances is that nature then selected for the macromolecules that gave that thing, let's call it the beginning of a cell, maybe some sort of advantage in its environment. So go ahead and try to answer this question. Press pause. This question is from the College Board. You can see down there, they're justifying why they wrote the question. There's all their justification. Oh, students need to know this science essential knowledge and how to do this practice and so on. So the answer here is letter C in the middle. Well, let's talk about these cells. I've got the little stages that your book is going to claim occurred in order to make these cells up here on the board. So maybe with as Dr. Miller has demonstrated, we're going to get some sort of synthesis of small organic molecules. Here might be some that you might find in one of these sludges. And then if you were to get a bunch of those, and you can imagine the early ocean, if these things come together, then maybe, I mean, they're going to bounce off each other. But if there's enough energy there, because there aren't enzymes yet, maybe a bond will break and another one will break, and then they'll reattach. And we've actually found that you can put these little tiny monomers near one another, and they will polymerize and turn into polymers on hot clay or hot rock. So joining of these small molecules into macromolecules, and we've seen this happen. And so you can imagine that if you have an open mind, that these things with enough time and chance might come together, and you might start to get some of these longer polypeptides here, a bunch of atoms that are stuck together. Maybe some of them were advantageous in their environment and they survived. You can imagine if we got these type of lipid molecules that they don't like water, they're nonpolar, right? And so you can imagine that they're going to organize around one another. And this is something that I think that kids and myself can really kind of agree with, that we might get these kind of, they're called micelles, or when you get this structure of lipids that kind of create like this sphere, and they're just, all they're trying to do is get away from the water, and they can't because they're in water, but they're gonna do the best they can. And so you're gonna get a membrane-like structure. So this is very similar to today's cell membrane. And so evolutionary biologists who talk about macroevolution suggest that there was once kind of these early cell spherical-like structures. Okay, I'm okay with that. Oh, sorry. Experiments have demonstrated that protobionts could have formed spontaneously from abiotically produced organic compounds. Yes, when you put a bunch of oil in water, they actually do spontaneously form these little micelle things that are trying to get away from the water. I'm okay with that. For example, small membrane bounded droplets called liposomes. So you can see the lipids in there, maybe the beginnings of a phospholipid, bunch of phospholipids, can form when lipids or other organic molecules are added to water. Again, for the third time, I'm okay with this. Okay, now here's where the stretch becomes. Protobionts exhibit simple reproduction and metabolism. Okay, so if you have a glucose with a phosphate group attached to it, which does happen in nature, and one of those is by diffusion gets into one of these protobionts. If then it has some amino acids that maybe we'll call a protein and maybe we'll say is shaped like an enzyme, if that has also come in by diffusion, then a reaction can occur. And an enzyme similar to phosphatase, which is something that just takes off a phosphate group that we learned about in chapter 11, if you remember the little PPs, those little guys that are the protein phosphatases, they can take off phosphate groups, and you can see that then eject there as one of those purple arrows. And then you can have many of those glucoses together, maybe let's call it a starch. And then if you get another type of enzyme like the one that exists in your mouth that we call amylase, maybe that's going to break down the starch into disaccharides, which we call maltose, which is just two glucoses together. The point is that these just serve as kind of as a safe place for reactions to occur that are protected from the outside environment. There's a lot of chaos going on the outside, but like our cells, it's just kind of a bubble that's shielding metabolism from the rest of the world that doesn't want metabolism to occur in an organized way. So we've seen this happen in the lab. They maintain an internal chemical environment. If you have an open mind, maybe this could have happened in the long ago primordial soup. And we start to get more and more reactions, and you know that life is just a bunch of biochemistry. And so eventually we might get these kind of cells, but it's important then, and this is an important point, number 4, is that, okay, great, you got one. Who cares? Because then a meteor comes down and blows up the whole area and the thing is gone. So you have to be able to reproduce it. And so there has to be some sort of code of information that says, okay, let's make another one of those. That thing seems to be doing okay. Sorry. The magic of the world is saying that thing is seemingly doing okay in its environment, and it's not being broken down by all the forces that are trying to break it down. But we need to make another one in order for it to really get a foothold here. And so there has to be some sort of information that's going to be passed from one to another. And that's where the origin of self-replicating molecules comes in. I want you to be thinking of information like DNA or RNA. So there's always been this question amongst evolutionary biologists: was it DNA or was it RNA as far as the first informational material that was passed from one precursor of a cell to another, like a protobiont to another? Your book is going to make the argument about is that it is RNA and not DNA. And it's not only because RNA is simpler, but there is another reason as well. Early protobionts with self-replicating catalytic RNA might have looked, okay, so the RNA might have looked like this. So the idea is this: you know from our genetics unit that RNA is single-stranded, so that's simpler. And number 2, they can create ribozymes. That's what we call them today. And the idea is that there's RNA can fold back on itself and then create these wild structures. And you can imagine if there was a pocket in there similar to like an enzymatic pocket in a protein, other molecules could get in there, they could somehow fit in there well, and then the RNA can twist and contort them so that their bonds can be broken a little bit more easily, and then maybe they'll reconfigure with other atoms to make something new. And so the idea is that when this says catalytic RNA, the idea is that RNA can act like an enzyme or a catalyst. You see all these different pictures of RNA structures, and you can imagine it's almost infinite what RNA can turn into, and you can have other molecules getting into these tiny little spaces. This catalytic RNA would have been more effective at using resources and would have increased in number through natural selection. I agree with that statement. That's it. We're done with 25.1. Thanks for your attention, and thanks for having an open mind.